immunotherapy options. This one slide is actually the answer of many questions. I believe you should be able to understand. मैंने सिर्फ एक पे डिटेल से बात नहीं की, that is about the biological response modifier. So there is a signaling pathway doing something. If you interfere that signaling pathway, you can block the function and the cell will start doing something else. Okay? Do ask me more about these things. And this is one example of how we can use a specific antigen which is present in cancer only to treat the cancer, to educate our own dendritic cells. This is some of the examples of the biological response modifiers. If you want me, I can speak more on this thing, but this is like an easier version. Okay, this is another example of the most recent and advanced drug that how it functions in case of HER2 positive breast cancer. Okay, I'm sure that you should be able to understand. I will make sure that aap coaches understand kar sake. Okay, now we know that in case of breast cancer, you heard that the involvement, you might know, you guys understand actually, the involvement of ER receptor, the involvement of PR receptor, and the involvement of Her2 receptor in case of breast cancer. If the breast cancer is because of the overexpression of the estrogen receptors on the cell, it is called as ER plus or ER positive. When the pathologist analyze the cancer cells and he let the patient know that he or she is ER positive, that means the cancer cells are carrying lots of estrogen receptor. And that is the reason of cancer in this case. It might be the PR positive, it might be the HER2 positive. I should teach you about these things as well, otherwise it makes no sense. Immunotherapy is more important for the therapy. It is more important to understand the problem, I can tell you. So in case of breast cancer, the breast epithelial cells, the breast epithelial cells, they carry the different types of receptors on their surface. One of the receptors is called as estrogen receptor. It's a protein. It's a protein. The job of that protein is to respond to a stimulus, and that stimulus is called as estrogen. During the menstrual cycle, the ladies, they produce like different levels of estrogen in their body. The job of the estrogen, it actually helps the cells to feel that I need to grow, I need to divide, I need to proliferate. So there are different cells present in the body which are required to proliferate at a certain period of time, and that is controlled by the estrogen. So the estrogen, once its level is increased in the body, its level is there. The estrogen binds with the estrogen receptors. So this is the estrogen receptor. The estrogen binds with the estrogen receptor. This is the E and this is the ER, and this is a normal type of breast epithelial cell. This is a nucleus. So this connection will activate the estrogen receptor, and that triggers a signaling cascade. It passes on the information to the next member, that passes on the information to the next member, that passes on the information to the next member. This member is allowed to enter into the nucleus, and within the nucleus, if I zoom in for you, within the nucleus, this is the nucleus, that thing is attached itself to the target genes. And those target genes will start activating, producing the messenger RNA. This messenger RNA will going to produce the proteins. You know, eventually it comes out, it start producing the proteins, and these proteins will start executing the function. And you know what is the function? The function is the cell to choose to go for cell division, means cells proliferation. So that is how the estrogen from the outside is able to trigger and help the cells to feel that I need to go for the cell proliferation. That is how it happens. It's not only the single cell, single gene, I can tell you. There are multiple target genes of this thing. But this thing is created out of all these series of reactions triggered by the estrogen from outside the cell. Now imagine if because of some accident, there are a lot of accidents, a lot of dysregulations can occur within the cell. One of the dysregulation, the most common of, is that a cell, for example, is supposed to produce 3 estrogen per cell. 3 estrogen receptors per cell. But what happens that accidentally the cell started producing more estrogen. It started producing more than normal number of estrogen. So let's suppose in a bad situation, the cell is overexpressing estrogen receptors. So now it is producing more than normal number of estrogen receptors. You can guess what? This cell is now more sensitized to the estrogen. this cell is now more sensitized to the estrogen. So for this is the less sensitive cell. This is now a more sensitized version of the cell. I mean, it will react quickly because of the presence of large number of estrogen on it. This is the thing which the pathologists see in the cancer cell. जब वो laboratory में भेजते हैं ना, when we send the sample to the laboratory, we actually see we label the cells with the antibodies against the estrogen receptor, and it colors the cell, and we came to know, oh, a lot of estrogen receptor is present on the cell. And such cell is called as ER positive cell. And such breast cancer is called as estrogen receptor positive breast cancer. So this is going to be more sensitive. That means it will going to react quickly, and it will going to proliferate quickly. But you know, we have a reason. We have a reason. We know the reason behind high level of cell proliferation. So we can interfere with the reason. What is the reason? The reason is the connection between estrogen and the estrogen receptor. If you block this thing, there is a drug called tamoxifen. If you block this thing, so with something, if you're able to design a molecule, for example, if you're able to design a molecule, actually this time we know the reason. The reason is that there are more estrogen receptors, so they are more sensitive. The cell will quickly respond to the estrogen, and they will divide faster, and they will going to develop into the tumor. So if we create a molecule, for example, if we can create a molecule whose structure is somehow similar and which can mimic the estrogen, so that molecule is like tamoxifen, which has developed by some pharmaceutical industry. So they give the person, the patient, the treatment with the injections. They send a lot of molecules of this tamoxifen, which is structurally very similar to the estrogen. It competes with the estrogen, and it binds with the estrogen receptors over there irreversibly, and that block the estrogen receptors in these cells. So you can sell once the tamoxifen is there, it will block the estrogen receptor, and cells will not going to proliferate in the same way. They will stop, and this is like a treatment of the cancer. So it is somehow a good news if the doctor tells a patient somehow cancer is not a good news, but comparatively, if the doctor tells that the person is ER positive. So what that means, in an easier word now for you, that we know the reason and we have a treatment. So now you should be able to understand what is the difference between what is meant by ER positive breast cancer, what is meant by PR positive breast cancer, and what is meant by Her2 neo or Her2 positive breast cancer. So if the cancer is like this, that means that it is because of the overexpression of estrogen, progesterone receptor, or Her2, human epidermal growth factor receptor. But if a cancer is like ER negative, or it is PR negative, or it is Her2 negative, that means it's a triple negative breast cancer. So then it is not because of the overexpression of these things. I mean, that's like a very, 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 very dangerous or sad situation, because we don't know the reason. So that is why such cancers are supposed to be very dangerous. And in that case, doctor started feeling that I might not be able to save the life of the person, because we don't know, we don't have any drug. Kisse milaye, kisse treat kare, we don't know the reason of the proliferation. It's a sad news. It's a bad news for the patient. Okay, so now let me show you. G.
PR positive.
I'm not getting your point.
What's PR positive?
Progesterone receptor. Okay. So the story will be similar. मैंने बाकियों की अलग-अलग explain नहीं किया, but estrogen receptor की तरह की story होगी. It makes sense? ठीक है?
Yes, sir.
Okay. Okay. You can give me your thumbs ups to let me know that all of you are alive out there. मैं ghosts और zombies को नहीं पढ़ा रहा हूँ. Okay? कभी-कभी मुझे पता लग जाता क्लास में. जी जी. Any question?
अच्छा सर, अभी आपने जैसे बोला कि ER पॉजिटिव जो है उसका ट्रीटमेंट भी है, और ये जो है हमें पता भी है किस तरह होता है, एस्ट्रोजन की जो प्रोडक्शन है वो ज्यादा हो जाती है, तो नेगेटिव वाले में क्या कम हो जाती है?
नहीं, मैंने ये बोला ही नहीं नेगेटिव वाले में कम हो जाती है, और ना ही मैंने इसको सजेस्ट किया। और एस्ट्रोजन की रिसेप्टर नहीं बढ़ती, इसकी प्रोडक्शन नहीं बढ़ती, एस्ट्रोजन रिसेप्टर का लेवल बढ़ जाता है। ओके, बी वेरी केयरफुल एज ए रिसर्चर, एज ए थिंकर, एज ए अंडर ग्रेजुएट स्टूडेंट। ओके, यू डोंट हैव वेरी मच एनी मार्जिन, क्योंकि जब आप प्रोफेसर से कम्युनिकेट करेंगे आने वाले दिनों में, यू नीड टू बी वेरी स्पेशल इन योर कन्वर्सेशन, एक्यूरेट, ऑनेस्ट। एस्ट्रोजन का लेवल नहीं बढ़ता है, इट्स इंक्रीज इन द एस्ट्रोजन रिसेप्टर, वन थिंग। ओके, क्या चीज बढ़ती है? लुक एट दैट, क्या चीज बढ़ती है? Estrogen receptor. That's
Estrogen
important.
receptor.
Okay. The next thing is that what happens in case of ER, the pathologist is looking at the sample. He stained it with antibody against estrogen. He did not find any staining. That means he did not see. He is not able to see lots of receptor. So that means he will label ER negative. उसको नहीं पता कैंसर क्यों हो रहा है. ये डॉक्टर का काम है. बाकी लोगों का काम है हिसाब लगाना कि अगर एस्ट्रोजन रिसेप्टर की वजह से, इसका मतलब ये हुआ, that means whatever the reason of the cancer is out there, that is not, this one is not the reason. This something else is going on. Okay? That does not mean estrogen receptor is not there. That means the estrogen receptor is not overexpressed, and that is not the reason.
So
Now, the most latest of the advancement we have done in treatment of breast cancer is the development of a molecule against which can bind with the Her2 receptor. Remember, there are a lot of people who are Her2 positive. If some people are ER negative, they are reported as PR negative, but they are reported as Her2 positive. That means still we have a chance to deal with those cancers, because we know that there's one of the reason might be there which is contributing in their cancer. So Her2 receptor is like it's a receptor, but like every other receptor, receptor is present on the cell membrane. This is supposed to be the cell membrane. Some part of there are other growth factor receptors like EGFR and Her4 and Her3, and there is one of the receptor which is called as Her2. I'm talking about this one. So this is the cell membrane. Some domain is outside the cell membrane. It should receive a stimulus. There is a stimulus जो Her2 को activate करता होगा. This is the natural stimulus which should go and bind with this receptor. Once it bind with the receptor, natural stimulus bind with it. I mean, मैं naturally बना देता हूँ. For example, this is a Her2 receptor. Okay, अगर मैं इसकी drawing अच्छे से ना बना पा रहा हूँ तो. So Her2 receptor is supposed to bind with the stimulus, and then it triggers the reaction in the same way. जो मैंने आपको बताया था, it will activate somebody. This will activate another one. This will activate something else, and that enters into the nucleus. And what will happen? It will go and bind with the target genes and activate the genes, produce messenger RNA, produce proteins, and start doing the function. And what will be the function? Cell will go for cell proliferation. Natural function.
Sir.
G.
Sir, one question, sir.
Sure.
Sir, ये मेडिकल बायोटेक के अंदर कैसे इंक्लूडेड है? मतलब ये तो कैंसर हो गया सर पूरा.
हाँ जी, बिल्कुल नहीं इंक्लूडेड है. कैंसर इम्यूनोथेरेपी इंक्लूडेड है.
अच्छा, इम्यूनोथेरेपी इंक्लूडेड
है जी. कौन साहब है ये? अहमद
नहीं.
हाँ, अहमद समझ आ गया. मेरा प्रॉब्लम
आ गया सर.
मैंने आपको इसका सोल्यूशन बताना है मेडिकल बायोटेक्नोलॉजी में.
अच्छा, ठीक है सर.
तो सोल्यूशन का पता लगेगा अगर हमें प्रॉब्लम को समझ आएगी. इज दैट राइट?
ठीक है.
जब मैंने आपको बताया कि एस्ट्रोजन रिसेप्टर की वजह से कैंसर होती है, तो आपने सोल्यूशन निकाला ना उसका, थेरेपी का. एंड व्हाट वाज द सोल्यूशन? द ड्रग मॉलिक्यूल व्हिच ब्लॉक द फंक्शन. इट मेक सेंस टू यू?
यस सर.
ओके, गुड. डोंट वरी. मैं खुद नहीं चाहता, मैं खुद चाहता हूं मैं तो थोड़ा सा पढ़ाऊं. डोंट वरी. ओके, मेरे से ज्यादा
नहीं सर, थोड़ा सी की बात नहीं थी. मैं तो आई वाज जस्ट वंडरिंग के
नहीं, मेरा
कैसे ये
मैं एक्चुअली एज ए इंस्ट्रक्टर आई नो and I believe that you cannot solve the things if you don't understand the problem.
Exactly, sir.
Okay, so if you know the problem, whatever I am telling you at the molecular level is the most latest and advanced things going on. And what is that? हमारा इसमें मेडिकल बायोटेक्नोलॉजी क्या है? मेडिकल बायोटेक्नोलॉजी ये है कि किसी इंसान ने सोचा कि एक मॉलिक्यूल बनाते हैं, look at this. और ये क्या करेगा जाके? ये Her2 को बाइंड कर देगा,
ब्लॉकिंग करेगा.
Okay, अगर Her2 ब्लॉक हो जाएगा, that means it will not able to send the signal, it will not going to activate the genes, it will not push the cells to go for the cell division. So if you don't understand the Her2, if you don't understand the reason why it is contributing, what the hell we will be doing with this thing? Okay?
Yes sir.
चिल है?
Yes sir, चिल है.
सदके जाओ, मेरे से ज्यादा कोई है ही नहीं. मैं तो दुनिया मेरे से ज्यादा कोई लेजी नहीं. डोंट वरी. मेरा जॉब है, प्रॉब्लम मुझे मजबूर करता है कि मैं आप लोगों को ये सारी बातें बताऊं. अदरवाइज इट सीम्स टू मी लाइक क्या कहते हैं, लेस डूइंग लेस फॉर यू गाइस. एनीवे, सो Her2 के लिए हमने ये सबसे एडवांस मोस्ट रीसेंट मॉलिक्यूल जो हमने डेवलप किया, और आपने ये सुना होगा आप में से बहुत सारे लोगों में जिनका ट्रीटमेंट होता है. इफ यू कैन रिमेंबर, दिस इज द टेक्निकल नेम ऑफ द ड्रग which is able to bind itself with the Her2 receptor. आपने सुना होगा कि वो एक इंजेक्शन लगता है ब्रेस्ट कैंसर के पेशेंट्स को, 1 लाख रुपया, 1.25 लाख रुपए का है. कभी नाम सुना Herceptin का? Herceptin. It's इंजेक्शन. अल्लाह ना करे आप लोगों को खुदा ना खास्ता कोई ऐसा केस हो तो फिर you came to know about. डॉक्टर सजेस्ट करते हैं Herceptin इंजेक्शंस के लिए. वो इतना एक्सपेंसिव क्यों है और वो मॉलिक्यूल्स फार्मास्यूटिकल इंडस्ट्रीज को आसमान पे क्यों लेके जा रहा है? क्योंकि दे हैव डेवलप्ड दैट मॉलिक्यूल व्हिच कैन परफेक्टली बाइंड विद द HER2 रिसेप्टर. अब वो इसको बेचते हैं ड्रग करके. तो ये एक ट्रीटमेंट है. बट what we are doing, what I told you, that apart from this type of treatments, the best thing of treating the best way of treatment of the breast cancer is to use our own immune system. ये बेहतरीन तरीका है, because इसमें we empower our immune cells, what we call as cancer immunotherapy. The things which you have studied with me is about all these possible options which we can use and utilize to kill the cancer cells. But you can see the problem which was asked by another student at the start of the class, if you can remember, somebody asked a question at the start. Okay, the trouble is that the cancer cells we are talking about, a lot of cells are normal, they are living happily, comfortably, and then what happens that out of all those between the normal cells, some of the cells started dividing rapidly faster, and they will going to develop a mass which we call as the tumor. ये एक issue है. This is the thing we are struggling with. Now the point is that if a cell choose to go for cell division, and if you remember all the things we have studied earlier, that the cell is in the state of push. It does not see that it is bypassing the cell cycle checkpoints. So the daughter cells will not going to be bona fide cells as compared to their parent cells. They will develop like a trouble. Let's suppose I am referring the trouble as one dot, and that trouble arises because of the cell cycle checkpoint, because of the hallmarks of cancer, or because of anything. That push will going to help these cells to produce more cells, and maybe in the meanwhile they will develop more troubles, or I must say more mutations. So they will develop a different type of mutations. They will develop even a different type of mutations. So at the end of the day, in this tumor, if there are different types of cells, they will not be like same. They will be like different types of cells. Some of them are very close and similar to the normal cells. They are fine. Some of them are a little bit different. Some of them are very much different from the normal cells, and they are the troublemakers. How you will be able to kill those things? How you will be able to kill those cells? The first thing is that the immune cells which are passing by, which are crossing, यहाँ से गुजर रहे हैं. So this is the T cell. Okay, मैं इसको एक अलग शक्ल देता हूँ. This is the T cell. It is passing by the tumor. It will feel this is normal, this is normal, this is normal. It should feel this is the bad thing. This tumor cell should feel this is the bad thing. This is the bad thing. This is not supposed to be here. ये जो immune cell है, this immune cell should be able to distinguish between the normal cell and between the cancer cells. But most of the time, the tumors, the immune cells fail to distinguish between these two. And as I have explained, because they have almost similar protein structure, DNA structure, and most of the phenotype are same. But if a lot of immune cells are passing by, not the one, some of the immune cell, this one brave little immune cell is able to distinguish this thing as a bad thing. Oh, this is not nice. Started interacting with it. That means in immune terms, that means it get activated. It get activated. If these cells get activated, what that mean? It will start killing that cell. But usually we fail. The reason of failure of our immune system is two. Number one, because of the number of immune cells which are able to know the trouble. The number of immune cells, there are very few cells who are able to distinguish between these two. And the second thing are other factors or other reasons that they are not able to create the exact response or the exact answer of the trouble. This is another technical reason that we fail to kill the cancer cell. How we can help our immune cells? That's the cancer immunotherapy. We will pick those cells out of the tumor. हम इस cells को जिसने पहचान लिया है, we will take this cell out of the cancer cell. We will grow this cell into the test tubes or in the culture dish. We increase its number, and then after increasing and raising its number, multiplying it, we will put that thing back into the person's body to increase the number of those immune cells which are able to kill the cancer cells. That is how we might be able to kill the cancer cells, by increasing the number. This is called adaptive cell therapy or the cell therapy or
Excuse me,
cancer.
sir.
GG.
Sir, the antibody that we are using to treat, for example, breast cancer, is that also taken this way, like the combination or the
No, 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 no,
information for
no.
that antibody?
No, no, no. This thing is developed by the pharmaceutical. All these things are treatments.
Synthetically.
हाँ,
Okay.
उन्होंने इसको स्ट्रक्चर को स्टडी किया होगा, ठीक है? इन्होंने इसके प्रोटीन
Yes.
स्ट्रक्चर को और फिर उसके बाद उन्होंने स्टडी किया होगा which molecule fits perfectly here. फिर उन्होंने ये मॉलिक्यूल क्रिएट किया होगा, thousands of molecules बनाए होंगे and eventually ended up in that one molecule. And this is the easier way. ये आसान नहीं है, बहुत easier way to
Yes.
find out who knows. This is the one, this is the hero who knows the cancer. So how about helping that one thing, increasing its number, taking it out, increasing. What is the other thing? The other thing is that if most of the cells are not able to figure out that this is the cancer, how about helping those cells to know more? How you can help those cells to know more? कैसे help करेंगे cells की to know more? If you want to help those cells to know more, look at that slide. You will find the specific things present in the cancer. You will find the specific antigens present in the cancer, and then engineer the cells. Educate the immune cells, so you will develop more immune cells which understand the cancer, which understand this thing. So what is the other things? Take out those cells. Take out those cells, put them into the tube, engineer them, let them know about the cancer, make them the perfect weapon, and then send it back into the person's body, and it will start killing. That's another way. So all of these things are different ways of treating cancer. Okay, so there are different forms. Some are called as general way of treating the cancer using our immune system. The other are called as the more specific ways of treatment of cancer. The general ways are what? The general ways are what? The general ways are like activating all the immune system. So if there is a cancer out there at one place in the epithelial, how about dropping some bacteria there? How about dropping some bacteria at that place? If you drop some bacteria there, what will happen? More of the immune cell will rush to the site. If more cells rush to the site, they will be able to, the chance of detection of the cancer cell will increase. So that is how you can help your immune system to detect it. You can work on this front by activating the immune system. Just 2 to 3 minutes. मैं ऐसे conclude कर रहा हूँ, okay? Just wait for 2 to 3 minutes. So one thing is that you should be able to increase generally the immune system. अब generally immune system का pressure जो उसका level up कर दें. And the last thing is that you can specifically engineer and specifically fix the problem. So we have addressed all those things in this direction, in these slides, and I'm sure they will going to help you to understand these things better. If you have any question, please feel free to ask to me in the upcoming classes.